Susie,
Rob,
how did you spend the 80s?
wearing bad clothes, leggings, pixie boots, big fan of pixie
Yeah.
boots. Running around outside in the street,
Well,
playing
running
that
around
kind of
outside
thing.
in the street is what I wanted to ask you about, I guess, as we would often go and play in the traffic. I mean, in the 80s, this is a really tired old trope of the kids from the 80s having to go around roaming the streets, but it's now a joke because that's what happens on Stranger Things, right? We had to go outside and play out in front of the road in the cars and huff that petrol fumes and lick lead, all those lead-lined fumes and the lead-lined plate. Have you ever stopped to wonder how much that lead exposure has damaged us?
I haven't, but I've got a feeling that I'm going to be thinking a lot more about it after today's paper. Is that right?
Yes, because I've now come to the conclusion that our generation may have been screwed over by this lead poisoning to a degree, an alarming degree that I hadn't really considered until today. So, Susie, what have you been up to since last week?
Well, I had a nature paper out, Rob.
Hey, I noticed. Well done. First authored Dinosaurs of Europe paper. Do you think we should do that for? I think maybe we should do it for fossils.
I d
You're a bit
We did my last one. I don't know. I
right.
mean, I'm always happy to talk about me, as you know.
Uh-huh.
But let's see how it goes. Let's see how it goes.
Okay, well, we'll put that. I'm interested in you telling me about these European ceratopsians. Is that right?
They are, yeah. Didn't you have
Yeah.
a paper out last week as well?
No, no, it's
Has it
coming
not
out
come out
next
yet?
week.
Okay,
Not
coming
come
out
out
next
yet.
week. Okay.
Yeah.
Okay, good. I'm
But maybe
glad I
we could
didn't
do
just
a
miss it.
no, no. Maybe we can do a you and me double hitter. But we wouldn't have to worry about this if you didn't have so many nature papers coming out.
Sorry, Rob.
So, but that's all right. So you don't need to
Well,
apologize
don't
for that.
worry, because I've decided that since I had a nature paper out in the second week of January, I'm going
Yeah,
to take the rest of the year off.
wise.
Mm,
Not before you've recorded some fossil files, though.
of
So
course not.
this week I've chosen a paper which has sent me down a bit of a mad rabbit hole. So each week we take it in turns. Susie and I discuss a particular paleo paper and bring it to your attention that we might find interesting or revealing. This week I have chosen, let me find the title, The Impact of Intermittent Lead Exposure on Hominid Brain Evolution by Renard Jonas Boao and Alison R. Mutrio and a whole bunch of other colleagues, lots of colleagues from South Africa, China, but mainly Australia, I think many of these colleagues are from. And this is a weird paper that combines fossils and embryology and genetics to look at how humans have evolved. So testing some hypotheses.
Now,
Was
once
it?
again, Rob,
Yeah.
with a paper that you have selected, I'm
Yes.
going to quibble with that word paleo here.
Yes.
A
Yes.
paleontology paper. Once again, I
Yes.
would argue this is strongly the overburden. But okay, well, we'll go on. I'll play the
Oh,
game.
I thought you were going to argue with me. Not, I thought that the paleo credibility of the first part of the paper was very solid. I thought that you were going to complain that the second part of the paper is completely not paleontology and is somewhat overwhelming. But no,
That too.
you're complaining that it's too recent. 2 million years ago is not the
Oh, come on.
the
You know
dire
you've
wolves. I
got
might
pants
accept.
older than that. You've got pants older than these fossils. You know you have.
Okay, so we're dealing with a story of human evolution here. So, which is relatively recent, but we're going back 2 million years, a bit older. But yes, it's relatively recent in comparison to lots of things we've done. Not as recent as the dire wolf, though. But
That's true. That's true. Yeah.
well, because as we know, the dire wolves now walk among us. Well, anyway, let's not get into that. So, this paper, the context of this paper is lead poisoning is something that's quite damaging to people, but it's often assumed that lead exposure is a relatively recent thing in the sort of urbanization age of humans, from sort of Roman medieval times up to the industrial period, where lead poisoning was ubiquitous, and especially during the 80s, where there was lead everywhere, apparently, and this has a lot of damage to people. So they wanted to take a deep time perspective on this. Now, I wanted to go off on some side notes before we get started. Are you ready for some deviations?
Absolutely.
Because that means less time talking about genetics. So presumably you're happy with that. Okay, so let me take you back then in this side note to Thomas Midgley Jr. So this is an American chemist. He's working for one of the car manufacturers, and he found a way of adding lead to gasoline that made engines breathe much smoother with an anti-knocking measure. Great, well done for him. Now, what he didn't know is that that was going to go on to create absolutely a public health disaster. But this guy is really interesting because Thomas Midgley Jr., he was responsible for creating it. Now, I can kind of forgive him. No, I'm not sure I can forgive him, but the point was he didn't necessarily know what he was doing. But after he invented anti-knocking lead additives to gasoline, do you know what he did next?
I'm going to imagine that he did something absolutely awful. Did he invent chlorofluorocarbonates and destroy the
He
ozone
did.
layer?
Did you actually guess
Woo!
that?
Yeah, that
No,
was a total guess. I did.
he did actually invent CFCs. You must
Oh
have
my
known that.
God.
You can't
No,
have
I
possibly guessed
didn't.
that. Right. Okay. So this is another
You
Children
set
of
it
the.
up. You set it up so that it
Yeah, I
led
set it
to.
up. So this is another children of the 80s thing. During the 80s, this was much on the 90s, there was much discussion about the hole in the ozone layer. So we have an ozone layer to protect the Earth from UV radiation, and it was discovered that there was a massive hole in it at some point in the 70s and 80s. And that was largely because of CFCs, which were created as a chemical that's highly inert and is used in refrigeration. Freon is what it's called, and it's been used in refrigerators and freezers, and it had to be banned. And actually, it's an enormous public health success because banning it worked and the ozone is now recovering. So that shows
It's
the power of these international collaborations.
it's slightly annoying, isn't it? Because there's lots of kind of right-wing commentators who claim that this is an example.
Yeah,
The hole in the ozone layer didn't cause all the hazards that everybody said it was
yeah,
going to cause, and
yeah,
thus climate change is the same. And actually, what they don't realize is that it's because we solved it. It's because
yeah.
science literally solved the problem, and the hole in the ozone layer closed up.
Similarly with acid rain, right? Acid rain
Mm.
and the whole of the ozone layer. These are things that we were talking about a lot during the 90s. We solved it. We got together. We did it. It's an example of how we can potentially solve problems. Anyway, what I find absolutely crackers is that the same person is responsible for lead in gasoline and CFCs and the destruction of the ozone layer.
It's amazing. What a guy.
So, sad postscript. In 1940, I was trying to think what happened to this guy. He never lived to know what happened to him because in 1940 he
Probably
contracted
did.
polio. So that's sad, which left him largely bedbound. Then in 1944, he died because he made himself a device to get himself in and out of bed using a series of pulleys of ropes, and it killed him.
Oh
So
my God.
this guy's. I'm laughing. I couldn't even. I'm laughing while saying the word kill. I can't get over the fact that this guy is such a disaster zone. He just he's poisoned millions of people, destroyed the planet, and then died in his own polio-ridden bed. Oh, good Lord, poor guy. Anyway, I shouldn't laugh so fiercely at that. So moving on, right? So modern estimations is that because lead has been added to the gasoline, it's everywhere. That the intelligence and mental health of children has been adversely affected. That there was, the cumulative drop in IQ points is 765 million IQ points have been lost cumulatively from children. And I think about
Rob,
this, yes,
imagine how clever we would have been if we hadn't have spent all that time. I'm thinking about this lamppost that was in my street that used to
yeah,
be where we played, like it was like home when we were playing 40
yeah.
40 and hide and seek. If we hadn't lit that lamppost so much, imagine how clever we would have been.
I'm not sure
Oh
I
my
can
God.
label. I'm not sure the lead in paint. I'm not sure it's his fault, but the lead in petrol fumes is the one that is his fault. You licking a lamppost is kind of on you, I think. I don't think we can blame Thomas Migley Jr. for that one. Anyway, so also this is associated with all sorts of horrible outcomes like ADHD and mental health problems. And when I looked at a graph in one of these things, it's like an enormous graph of not so much a problem for the boomers, then all the kids of the 80s, and then it goes back down again as they fix the problem. So you and I are right in that danger zone of having been messed up by lead. And also violence. Apparently this leads to public disorder and violence and crime.
Okay. Well, that explains many of my feelings then sometimes.
There you go. Yes. Well, I mean, your rage problems may or may not be related to lead. I don't know. So we're getting a bit too deep into diagnosing each other there.
Do you think I
So
could use this, next time I get hauled up in front of HR for the
just lead.
behavior and respect policy? I'm sorry, what can I say? I was a child of the
It's
80s.
just
It's lead.
lead.
It's lead's fault.
Yeah. So that's the context where this paper starts, right? We've got this. One final quote about Midgley. Fred Pearce, the New Scientist, described Midgley as, quote, a one-man environmental disaster, end quote, which I thought was kind of funny. Anyway, so this is the context of where we start. These authors therefore want to look at the ancient prospect, not just the modern, like, how has lead affected populations of people? They want to look at the context of lead in the context of human evolution. So they took a series of fossils of human fossils from Africa, Australopithecus, which we've mentioned before, I think, some early Homo, and some ones from France, some Neanderthals, which we definitely have mentioned, some Homo sapiens, and Gigantopithecus from China, and some other ancient humans from China as well, and some, other material like some baboons and some orangutans. So what they did, they took these teeth, they sliced them open, they had a look, and they tried to trace where the lead was concentrated in the teeth. And they found lead banding, especially in the dentine. So as the teeth were growing, they found evidence of lead associated with growth, especially from young ages of 5 to 6. But this is when the lead poisoning can have the most effect, is when you're children, when you're around 5 or 6. Now,
I have a
that's
question
kind of,
about this
yes,
because it talks about between 1 and 6 years and lead
yes,
being
yes,
particularly. Now, I don't know about ancient humans, but we don't have adult teeth between
that's correct.
one
Yeah.
and 6 years. So are these all baby teeth? Are they milk teeth?
Good question. I don't remember them mentioning that, but
Yeah, they
they
didn't. I don't think they mentioned it, but I was a little curious as to whether these were the teeth of juveniles or the teeth of adults because they talk about this 1 to 6 years time period, but they don't specify. But I guess the banding occurs throughout all of these teeth, regardless. So probably doesn't matter.
well, that's where I get a bit confused about this because this is the paleontology part of this paper. We probably should just spend a bit more time talking about it before we go into the weird genetics and brains in a jar. But we've got here, so they talk about these lead bands, but they don't. What frustrates me slightly is that they find lead everywhere, in the crown, in the bands, in the roots, in the enamel, in the dentine, in the humans, in the non-humans. So I don't really know what the control is here. Basically, there's a lot of lead, and they spend some time talking about diagenesis, which I think is important because that was my question here.
Yeah,
So, just to
exactly.
explain to the reader what diagenesis is here, if you've got a bone and we bury it and it turns into a fossil over time, great. And we can maybe look at the chemistry of that fossil maybe 10 million years later, a million years later, 100 million years later. Problem is that chemistry of that bone representing what was originally in the bone when that organism was alive, or does it represent what happened to that bone as it was buried? And all sorts of liquids can go through a bone and replace certain things in it. Like if you find arsenic in a bone, that doesn't necessarily mean it was there in the animal when it was alive. It might just mean that the water has flown through it and different chemicals have replaced different things. And that's one of the things that we have to think about a lot. Some of the research we do here in Manchester, where we're trying to do a lot of these chemical analyses. Now, they tried to approach this by saying, well, it's in the banding, it's in the interior. But lead is one of the more slutty chemicals. It can sort of turn up in all sorts of places and get preferentially replaced. I may have to come up with a different metaphor for that. So they compared it with uranium, and
Mobile.
they
It's the more mobile.
it's more mobile, shall we say? And because these things are buried in caves, it's quite possible that lead was just naturally going through these caves and suffusing these teeth. But they compared it with uranium, and they found that the uranium was sort of in very low levels, of course, but uranium is naturally occurring. And they found the uranium was sort of evenly distributed, whereas the lead was in these specific bands. That's their justification for this. But they did actually specifically say that there was some diagenesis. So I don't know how much this is original and how much is not.
I mean, that was my initial thought when I was looking at this. I was like, oh, come on, this is all diagenesis. How can
Hmm.
you determine? But actually,
Hmm.
I did find their arguments relatively compelling. I mean, I think a thing that I noticed in their cross sections of their teeth, you've got dentine and then you've got enamel. And a thing that we know
Yeah.
is that dentine is much more likely to undergo diagenesis and be altered, whereas
Yes.
enamel's very stable, isn't it? So if we were
Yes.
going to do isotope work on kind of deep time fossils,
Hmm.
we tend to focus on enamel rather than the dentine. And you
Hmm.
can see that here, right? In their figures, you can see that the dentine is much, much more enriched in lead
Yes. Yes, absolutely.
than the enamel is. But then the fact that the dentine is more enriched in lead, does that suggest that some of this lead is actually diagenetic?
Mm.
Why is it? Why is the dentine more enriched than the enamel?
Well, I wanted to see more on this in this paper. I wanted to see some more teeth. I wanted to see some controls. Like, let's look at some specimens that didn't have lead in them. Look at some non-humans. Maybe look at a cow or something. I don't know, something that's different.
Yeah, that's a good point.
Do you want to go on another side note about
Yeah,
Gigantopithecus?
let's
Yeah,
do
let's go
it.
on a side note about Gigantopithecus. So Gigantopithecus is a fairly odd one. It's not part of the human story, but it's a specimen. It's a species from China and I think South Asia as well. So Gigantopithecus was first discovered by looking up his name here, von Koenigsberg, who was a Dutch guy working in Java. So he's Dutch, but he was born in. So he's Dutch, but he's German. So he's ethnically Dutch, but he was born in German. Why am I mentioning this? Because it comes up relevant later. So he was working in the 1930s, finding all sorts of fossils. He found some teeth in a jar in a drugstore in Hong Kong and thought they looked weird. Turns out they were Gigantopithecus, which had never been discovered. So he was then collecting these specimens. He had the locals bring him specimens, but he told them he was going to I will give you money per specimen bought to him. So then the locals started splitting up the specimens into lots of different chunks. Then he was like, no, you can't do that. And then he said, you refused to work with them. So then they burned everything they'd collected out of spite. So that's great. And then when the Japanese started invading Indonesia and Java, this guy panics and put the Gigantopithecus specimens that he'd been working on, he put them in a milk bottle and buried them. And then he was interned in a Japanese prison of war camp and then went back to extract them afterwards and then went back to New York. This is just absolutely bonkers.
Well, it's a
This is
good job. It's
a
a good
weird
job he
story.
survived the war, because if he'd put all his fossils in a milk bottle, buried them, and then died, nobody would ever know where they were, would they?
No, and that would have been a confusing archaeological thing. So we've
Wouldn't
got
it?
these Gigantopithecus teeth in a. Anyway, so this is what is Gigantopithecus? It's a massive orangutan, right? So
Oh,
now
right.
the
Okay.
problem is we don't actually know how big it is because we've only got teeth and jaws. But the estimates are that it was about 300 kilograms,
Wow,
a
that is a
massive,
massive. That's a
yeah.
massive orangutan.
So yeah, absolutely gigantic orangutan type thing. And this one was a reason I mention it is because this was one that had the biggest concentration of the lead.
It did. Yeah, yeah, yeah.
Mhm. Okay, so let's move on to the next story. There is lead poisoning in these teeth. Now, we go on a screeching gear change at this point into an
Oh,
absolute mad scientist stuff here, which I won't spend 2 ages on because this isn't the genetics files, this is fossil files. But we have to mention what they did next. They start then going on about this gene called Nova1. So Nova1 is a gene that humans have and non-humans have, and Neanderthals and Denisovans have one version of the gene, which they call archaic, and modern humans have a different version of this gene. And that's often been implicated in growth and development and how it might be playing an important role in language and brain evolution and lead. So they looked at this one specifically. So they started by looking at how did this is the bit that I was a bit surprised about because, the shtick is I'm meant to be the biologist and you're the geologist, but this is a level of biology that's far beyond what I'm familiar with. They made some little tiny brains in a jar.
I mean,
So
they grew a brain. That was
they grew a
I
brain.
was
Yeah.
reading it and I'm going, when they say organoids, do they mean they grew
Yeah.
a brain in a
Yeah.
jar? Is that
Yeah,
what
yeah,
they
yeah,
did?
yeah, yeah,
I
yeah.
was like,
This,
wow.
this is some mad science stuff. They made these brain cortical organoids. So
Yeah.
cortical organoids, COs, which they refer to, using pluripotent stem cells. So stem cells can be extracted from all sorts of places. They can be, but they're really useful. They can be used for all sorts of things. And because they're called pluripotent, because stem cells can develop into any kind of organ or in the body. But these organoids are these tiny little blobs of cells that are made into a sort of cortical, a brain. These little brains in jars, and they're tiny. And this really, I mean, I was a bit surprised by this because they're very casually mentioning this, whereas I was like, oh my God, brains in jars. No, they're just tiny little balls of cells. I was very obsessed with trying to find out how big they are. They're a few millimeters across, I think, these tiny little things. And they made two versions of it, one containing the normal human gene for Nova1 and one containing the archaic Neanderthal type one, and then grew them
I mean,
up with.
genetics really has changed a lot since we were at school, hasn't it?
I know.
I mean, it
Yeah.
really has.
Yes.
It's really moved on. The idea that, first of all, you can just, you can grow a brain in a jar and then you can genetically engineer it to express
Yes.
a gene that we have identified in a Neanderthal. That's
Yes.
insane, right?
It is
That's
absolutely
crazy.
insane.
It's crazy. Surely we should
Um,
be able to solve all of the world's problems with genetics, or like some of them, maybe not climate change,
well,
but
I mean, as we,
diseases.
if people want to go and find out about how you can and cannot, and indeed emphasis on cannot solve the problems with genetics, go back to episode 3 or 4 where we looked at colossal biosciences, and we can have a look at
Uh,
the
okay, fair
yeah,
enough.
yeah, yeah. Now, I didn't realize this, but I did a bit of looking into this, and apparently there were some people that disputed whether you can or can't just create the archaic gene and put it into things. There were some
Ah,
people saying that this was a mistake, or you can get CRISPR errors. And actually, the reason that the gene behaves differently is because they basically just constructed it badly. But anyway, we can assume that these guys have solved that problem, even though these guys didn't cite that paper. Moving on, they grew up these little tiny brains in jars, which sounds crazy, with and without lead exposure, with and without the two versions of the gene. Then they looked at the morphology of these things. How did they grow? Unsurprisingly, they were different. Then they looked at the single cell RNA, which again, this is mad. When I think back to my biology education, what, 25 years ago, the idea that we could do this level of precision, take one single cell, look at the RNA that was expressed in it and the proteins, absolutely bonkers. Anyway, they ended up with 99,000 cells and then tried to classify them as the sort of things they were doing. Anyway, the point was the gene expression patterns in the two of them were different, not surprisingly. And they looked at these basic massive maps of the different gene expression patterns and the gene expression patterns different between, sorry, the gene expression patterns different between the human and the archaic human, and the gene expression patterns differed between the lead exposed and non-lead exposed ones. So that there is a big difference there. And they found that one of the big genes that was implicated is this gene called FOXP2. Have you heard of FOXP2 before?
Not prior to reading this paper, no.
No, so
It
I went
sounds
on a
like
bit
it's
of
important though, isn't it?
well, it did sound like it was important. So FOXP2 is very much varied according to the lead exposure. And FOXP2, I hadn't heard of this until I read this paper. It is a really important gene. It's called the language gene.
Hmm.
It's a gene that's involved in transcription and all sorts of things. I won't go into the cellular function of it because that's not particularly interesting.
Thanks.
But it's associated with language. What's the word I'm looking for?
Development.
Yeah, development language, and it's if you don't have the proper version of it, you could have all sorts of verbal dyspraxia, and it's not just about your ability to understand; it's about your ability to make words. So
Hmm.
it's about your movement of your tongue and your throat. And if you've got errors in that gene, then you might also have difficulties with coughing and sneezing and swallowing.
Huh.
And also, birds and bats have lots of important things to do with this gene associated with echolocation. So it's massive,
Oh,
and it's
really?
associated with ADHD, and it's associated. You can mess it up if you have too much cannabis. You can get errors in this gene. So this is really interesting gene, which they find to be closely associated with this. So in conclusion, so they observed that the archaic version of this Nova1 gene, the organized exposed to lead, led to a loss of the FOXP2 expressing neurons and the absence of distinct cell populations. So basically, you ended up with, if you had the archaic version of the gene and you're exposed to lead, you didn't develop properly, and you would have had a massive effect on your ability to use language.
But
So
also the
yeah,
archaic version that also affected FOXP2 differently, didn't it? So
yes, it did. Yeah,
so there's something already right about
yes,
the archaic version versus the human version
yes, yes,
about better social interaction and both better speech development.
yes,
So
yes,
suggesting that we have, more advanced abilities in that region anyway. And
yes.
then lead like amplifies that to some extent.
Yes. Yeah, I was just about to get to that. Don't worry.
Sorry.
No, you're right, though. You're right to highlight that. So then they say, quote, these data indicate that the archaic variant fails to buffer against lead-induced neuronal stress, supporting the conclusion that it conferred reduced resilience and thus a potential survival disadvantage under environmental lead exposure. In contrast, modern human variant offered relative protection against this neurotoxicant. So that's the end quote. Their idea is that because we as humans had this extra version of this gene, we were able to deal with the lead exposure, whereas the archaic humans were not able to. Now,
But we're not able to deal with it fully, are we? Because it still
well,
affects
no,
us.
still
But
not,
maybe
but
a bit
better,
it affects
better
us less.
potentially.
Yeah. Okay.
Now, what I found a bit frustrating about this is that that's obviously not the whole story because, they've got a model where they've taken a human model and they've put in this archaic gene and they've taken, and the gene has been messed the same. But actually, of course, other genes in an archaic human would be different. So it's not
Yeah.
really the same because the archaic version of that gene would need to interact with the archaic version of the other gene. So there could be some mismatch there between the two
Oh,
of
that's a
them.
good point. They do talk a little bit about their limitations of their tiny brains in jars, don't they? And they say that look, this
Yes.
isn't actually this isn't equivalent to an actual brain, and,
No,
and and obviously brains also interact with other parts of the body, as well. So
indeed. Absolutely. And that is a widely held difficulty with these little organoids is that they are not real models. And this is widely held as a limitation. So they're right to highlight that. But it raises
but
an interesting
also
question.
you can't, like we have agreed, I think, as a species that we're not going to genetically modify tiny human embryos to have
Hmm.
you know the DNA of Neanderthals just to
Hmm.
do experiments about fossils, right? So
Yes.
ethically, there's really no other way of doing this.
Yes, and this isn't really about solving human health conditions. This is
No.
mainly about looking at an evolutionary study. Although it did make me wonder what would happen, in an evil scientist kind of way, if I took a chimpanzee and edited it to put in our version of this Nova 1 gene. What? Well, we're going to end up with a chimp saying, "Hello, good day to you." What's going to happen there? But that's obviously deeply unsettling. Well, let's not
Unethical.
do that. So that was the conclusions of the paper. They're saying that the lead is the really important story in the origin of humans, in the sense that our ability to overcome the lead poisoning might be relative to the archaic humans, because we still can't. Might be related to language, might be related to development, might be related to all sorts of different things going
Well,
on.
social cohesion as well, right? This idea
Hmm.
that they said that lead poisoning in human populations is known to be associated with antisocial behavior,
Hmm.
and that maybe the selection for the modern human version of this gene allowed better social interaction, which helped us
Yeah.
survive in adverse conditions.
Absolutely. And again, the adverse conditions is interesting, because that's when the lead poisoning might turn up. There are all sorts of
Mm.
natural conditions. So we often talk about lead as in terms of urbanization and industrialization, because the Romans were also messing around with lead. But I don't know what they were. Maybe they were all mad, violent, crazy people because their water was moving through lead pipes. But the industrial processes and urbanization processes, but also naturally it can turn up in. They have a bit of explanation about this. Uh, Different patterns of lead exposure could suggest that Paranthropus lead bands were the result of acute exposure, for example, wild forest fires, whilst in the other two species that are known to have more varied diet, lead bands might have been more frequent through the bioaccumulation process in the food chain. So they're suggesting that basically lead poisoning might have been a fairly ubiquitous in natural environments due to things like that. Living in a cave is also going to make a difference, depending on how you get on with. But you raise a really good point about the social cohesion.
I also just a thing that I a rabbit hole that I started to go down but didn't have enough time to finish was I wondered whether there were actually, because what they haven't really done is link this to anything in the environment. So they've
Hmm.
speculated about sorts of things, but you might actually be able to. I mean, if you're an archaeologist, you might actually be
Hmm.
able to go and look at these cave deposits and
Hmm.
say, well, is there high lead in
Yes.
in the caves where these things were living? Oh, we
Hmm.
have the sedimentary layers where they're found.
Yes.
So
Yeah.
can we see
Yeah.
variations in lead, or something that could cause it? Can we see, is there
Hmm.
evidence in the sedimentary deposits where this animal were
Hmm.
living of
Hmm.
frequent wildfires? And one thing that I went to look at, because the ones from China,
Hmm.
the Gigantopithecus and Pongo had the
Hmm.
highest, from 2 million years ago, from the Pleistocene 1.9 million years ago, had the highest lead. And
Hmm.
I went to look whether there was anything. Is there anything? Because volcanic eruptions, right? That
Hmm.
if you have big clouds of volcanic
Yeah.
gases or volcanic ash, that would be a source of
Yeah.
lead.
They
And
specifically
were
mention
there?
volcanoes. Yeah. Yeah.
They do, yeah. And was are we seeing in some of these areas actual, you know time constrained volcanic eruptions at this time? I did
Hmm.
find a volcano on the Chinese Korean border that
Hmm.
was active during, still active today, but it was active
Hmm.
2 million years ago. um but I think that for me that would have been really interesting to actually
Yeah.
go and try and tie, because it is quite speculative, this, and there
Hmm.
are these issues around diagenesis, but actually being able to tie what the animals were experiencing in the environment to something that you can record in the environment. I mean, it should be possible.
Yeah, well, that's what you're the geologist, right? You want to look at the rocks and think about where the causes of this were coming from and the environment these things were living in. But I guess that also makes me think about the giant. I mean, if we can, it makes me think about diagenesis. If we can demonstrate that there was a lot of environmental lead, then maybe that has a biological effect. But it also certainly would have had a sedimentological effect. It would,
Yeah,
the lead-rich environment you find yourself in means your teeth might be more bathed in lead, especially if it happened after the organism was alive, right? If you think about that, maybe this Gigantopithecus in a cave, it dies, and then 100 years later there's a volcanic ash everywhere, then
yeah, it's
that
going to
makes
seep
no
into the
difference.
teeth.
Yeah,
Well,
exactly.
I did also wonder whether these growth bands, you sometimes get, I'm trying to think of an example. I mean, you certainly see it in rocks. I don't, I don't know whether it's the case in fossils. I don't know whether anyone's looked, but whether there's different porosities within
Mm,
specific growth layers.
mm,
And
mm.
thus, if one layer has a different porosity, it might be easier for water to move through it. And so the
Absolutely.
diagenesis might actually deposit in specific bands, and we see this in
Mhm.
rock. So it could be that the same thing could be happening in fossils, and actually the lead is picking out, slightly lower porosity
Yeah. Mhm.
growth bands in the enamel.
Absolutely. So I think there's a really interesting story in there potentially about what was going on with archaic humans. It's a really interesting example of looking at the interaction between geology, paleontology, evolution, genetics, brains, languages, sociology. I think that's a really interesting idea. Hard to test, very
Yeah,
hard to test. I mean,
yeah,
they
yeah,
talked about some other way that they were very focused on some more genetic ways to look at this. I'd be interested in looking at what was actually happening to the fossils.
yeah, exactly.
Should we have some concluding side notes? Side notes and then side notes on the side notes and then even more after that.
God, I love your side notes, Rob.
We've had a lot of side notes today. Side note, they declare that they've used AI in this.
Oh, I didn't see
Yeah.
that.
They
What did they
explicitly
do?
declared right at the very end, they say we used AI tool ChatGTP, GTP4 Turbo, developed by OpenAI, to assist in the curation and structuring of the GOBP terms using, that means gene ontology biological processes. The AI was used to filter, modify, and organize the geo terms provided by the authors according to specific criteria relevant to neurobiology. So, what that means is, in part of their genetic analysis, they had this big gene ontology, GO analysis, where they had to sort of interpret what all those genes were doing. Now, that means that they had to probably download hundreds of papers looking at these cell types, and they asked the ChatGPT to go through those papers and come up with a summation of what sort of type of, classifying those papers as saying what that gene was or was not doing, or what that cell was or was not doing, and what the function of it was, because that's important for your big maps. Now, are we happy with that?
Um, I mean, it's difficult to have a view on a subject I don't know anything
Yeah,
about. If
yeah,
somebody had written this in a paleo or a geology paper and said,
yeah.
I basically use ChatGPT to
Hmm.
summarize 8,000 papers
Hmm.
and tell me what they said,
Hmm.
I'd be deeply skeptical.
Hmm.
Having seen
Yeah.
some ChatGPT summaries of some of the literature, just make stuff up, right?
Yeah. So I find this quite interesting. I think this is difficult because I can't say much more about it. I just wanted to bring our attention to it
It's
because,
interesting, isn't
again,
it?
the gene
You don't,
ontology is an interesting one. It's basically it's a labor intensive thing to go through and classify these things. Uh, but I'm not in a position to know whether that would or wouldn't, what the biases it would
yeah,
be. Like, I think you're absolutely right. Like in the paleontological example, you and I talked about all sorts of biases that can come in because we did a whole episode on this for people that want to go and look at it. Well, we as paleontologists know about the structure of our data, the biases of our data, and we as humans deal with that all the time on a manual, physical, person by person, data point by data point. When it comes to this particular example, we're not in a position to know or understand what differences that could or couldn't make. I just thought it was interesting that they have explicitly stated that they've used ChatGTP to classify these gene ontologies.
hmm, yeah,
But
interesting.
this did remind me of a different thing, is that I'm a little bit frustrated. I mean, I've got to be careful about what I say about this because my biology education is 20 plus years old. But this did strike me as a little bit of a fishing expedition. We've got these two different organoids. We've looked at the gene expression patterns, and then after the fact, we've decided that we're interested in the language one. If you just look at all these, if you do a scattergun approach to all these genes, you can end up saying what you want about brain development because you're interested in brain development from a beginning point. You don't start by saying, I'm interested in this gene because it's results brain development, because you can't often. Genes are
Yeah,
often associated with all sorts of things. This gene ontology thing is difficult because one gene can do all sorts of things. Even with this FOXP2 one, it's involved in language, but also it's, expressed in your lungs, it's expressed in your throat, it's expressed in your brain. Genes don't do one thing. There's not this
yeah,
nice pattern where we have one gene does one function. Life would be so much easier if that was the case, but they're not. They're all interlocked. They're all doing different interdependencies. They get different splicing. Even one gene can have different functions depending on the environment it was put in.
yeah.
So this can really create a problem. Anyway, that was just one thing I wanted to go. Is it just a big fishing expedition? And they've ended up finding the genes that they were interested in and telling a story about that after the fact. But again, I'm not in a position to say whether it is or isn't the case. Side note on the side note, competing interest statements again,
Oh, haven't read it. Haven't read it. Where is it? Where is it?
right at the very, very end of the paper. So ARM,
Oh,
which is the corresponding author, Alison, whatever he
hmm.
was, is the co-founder and has an equity interest in Tismo, a company dedicated to genetic analysis and human brain or
Organogenesis.
organogenesis on therapeutic applications customized to autism spectrum disorders and other neurological diseases. ARM has several patents on stem cells and brain organoid models. The terms of this agreement have been reviewed and approved by the University of San Diego. So
California,
that's interesting.
University of
So this is,
California.
yeah, University of California. San Diego. So this is another example where the person doing some sort of genetics and bioinformatics stuff has has a financial stake in the usefulness and impact of what comes out the other end. Now they've been clear about it. They said they've been reviewed by the panel and it's all above board. But I still think that's interesting because we had the same problem with Colossal Biosciences, where the Colossal Biosciences paper said, "Oh, we are absolutely great at de-extinction. This is amazing and it's brilliant and it works." And actually, we own the company that does all this.
Yeah, I don't know that this one is as bad as that though, because
It's not as bad
what
as Colossal Biosciences, but not many things are.
no, fair. But what I'm getting from this, so Alison is the co-founder and has an equity interest in a company dedicated to genetic analysis and human brain
Yeah.
organogenesis. I mean, an alternative view, for argument's sake, could be
Yeah.
that actually, she's running a private company which is
Hmm.
extremely well resourced at a
Hmm.
time when science funding is difficult to come by, particularly if you're in the States, for example, or in some other places where right-wing governments have come in and stripped out science funding. Argentina is
Yeah.
another one, for example.
Yeah.
And actually, what she's doing is putting the facilities of her lab and some of the things that she's developed, like these organoid models, and allowing doing some work that allows paleontologists to come up with and do
Hmm.
some interesting, to make their work a bit more interdisciplinary. So it could,
Yes.
an alternative reading could be that this is, somebody who sits on the border between
Hmm.
academia and uh,
Yeah.
industry, I guess,
Yeah.
actually, allowing resources from their company to be used for kind of a relatively niche study into human evolution.
I think that's right. I think that's right. And it's a he, by the way.
Oh, he.
Confusingly, even though they're called Allison, it's a man, it's a Brazilian guy.
I
So
apologize,
yeah,
Alice.
I think that's right. So that's interesting. And then so I think one final thing before we go home to the take-home messages is that when I was looking into this, I came across an article on New Scientist where there was a quote by Tanya Smith from Australia. So Tanya Smith is from Griffith University, Brisbane. And it says, quote, she isn't convinced about the extent of lead exposure or whether the work on organoids can be extrapolated to an evolutionary advantage for modern humans. So this is now Tanya Smith quoting. This is a really complex paper that makes some highly speculative claims, says Smith. While it is no surprise to me that wild primates and ancient hominids were exposed to lead naturally, as we've published multiple papers over this over the last 7 years, the limited distribution, number, and type of fossils included simply does not demonstrate that human ancestors were consistently exposed to lead over 2 million years, end quote. So Tanya Smith has absolutely slammed this. So as a result of Tanya Smith's quote, I had to go and look up. And yeah, she has published papers saying that the lead exposure in Neanderthals was demonstrated very clearly already in the and when she spent a lot of time talking about diagenesis and exposure. Thoughts?
Actually, it's interesting because one of the thoughts that I had reading this was there's not very much context about the fossils, where they're
No,
found or how they're found or
no.
their taphonomy. And one of the things
Hmm.
I thought was, how do we know that these are normal?
Hmm.
So if this lead is real,
Hmm.
and we know that lead poisoning causes massive neurological problems, how do
Hmm.
we know that these aren't like massively pathologic individuals who
Hmm.
died
Hmm.
and are were found, I don't know, in the environments that they found were in the caves because that was where they crawled to and died, or because they were
Hmm.
abandoned by their social
Hmm.
group, or whatever.
Hmm.
So how, I guess, there's always, when you have so few fossils, how do you know that the one that you're looking at is the normal one? And this actually takes us back to the thing we were talking about last week about the hadrosaur mating and injuries and hadrosaurs. How do we know that we don't have the fossils simply because they're the weird ones, they're the odd ones
Hmm.
out, they're the pathologic ones? And so that did occur to me when I was reading it. And I guess what she's saying kind of is supportive of that we there's just hardly any specimens that they've actually looked at here.
Yeah, I think that's right. And I think Tanya's quote is really interesting. But I think it's also interesting because she's just really annoyed. Because reading between the lines, she's like, "Yeah, I said this 7 years ago, and
You
you
haven't cited
guys,
me.
yeah, okay." But that's what I did. I did go and look at. I looked it up. I went and read her paper that she wrote like 7 years ago, and I was like, "Oh yeah," she said she demonstrates these Neanderthal children had lead poisoning in bands in their teeth, and I was like, well, why didn't they cite it? So I went back to the paper and they did cite it, but they cited it, buried it deep in the methods, which I thought was a
Uh,
bit disingenuous, which I thought was a bit cheeky. They didn't mention it in the context of thinking about, the actual state of the place. So Tanya Smith is, she's been calling them out, and I wanted to amplify that, I guess. So that's where we're at. So in conclusion, then, we've got this important picture from the fossils. There's lead in bands all over these teeth from humans and non-humans and children and non-children in enamel and dentine. In fact, it seems to be ubiquitous. But they seem to highlight that may have played an important role in human evolution by saying that humans may have evolved this gene that allows them to escape the environmental burden of this exposure more and allow them to continue to have language, social cohesion, all sorts of brain development, which is an interesting story. But as you and I have highlighted, there's quite a lot of questions, shall we
Questions.
say?
Yep.
Future
Caveats.
prospects,
Yes.
caveats. Yeah.
Areas
So
for avenues for future research.
So I wouldn't say that lead, I mean, I was originally thinking that this is demonstrating that lead is what made us human, but actually it's not. It's more like the case that maybe we were able to cope to escape from lead, maybe more than the others, and become more clever. I do worry about us being children of the 80s and my brain rotting and having the attention span of a flea. But then I also wonder if our children who have grown up in a lead-free environment are really, I don't know if I can look to them as the genius children that will save us with high levels of concentration and superb mental health. I don't know. I'm not quite sure about that. The jury's out, I guess. We'll see. Right. Well, thank you, Susie. I'll look forward to you telling me about something a bit more fossily next time, maybe something a bit older.
Yeah, we'll talk about something older. I'll let
Yeah,
you know what that's going to be when I figured it out.
and I'm sorry for subjecting you to so much genetics. Bye.
Bye bye.
The Fossil Files was presented by Robert Sansom and Susanna Maidment. You can find more information, including links to the original papers that were discussed, at fossils.libsyn.com, and you can contact us at fossilfiles.pod@gmail.com.